Next, we will investigate how plasmolysis occurs in plants. To observe plasmolysis, begin by obtaining rhubarb cells or another suitable plant tissue, ensuring the sample is fresh and prepared for examination. You can use a microscope slide to mount the tissue, making sure the cells are clearly visible. Next, carefully add a 30% sugar solution to the cells or tissue. This concentration is high enough to create a hypertonic environment, meaning the concentration of solutes in the sugar solution is greater than the concentration of solutes inside the plant cells. As time progresses, observe how the plant cells react to the solution. You should notice that the cell membrane begins to pull away from the cell wall, which is a characteristic sign of plasmolysis. This occurs as water moves out of the plant cells by osmosis, leaving the cells shriveled and the plasma membrane detaching from the rigid cell wall. Record the changes over time, noting how the extent of plasmolysis increases with the duration of exposure to the hypertonic solution. This experiment provides a clear demonstration of the effects of osmotic pressure on plant cells and the process of plasmolysis. The results indicate that adding a 30% sugar solution to the rhubarb cells caused plasmolysis. The cells contracted and detached from the stiff cell wall, forming a noticeable gap between the cell membrane and the wall. And the conclusion can be drawn that the results support the occurrence of plasmolysis when plant cells are exposed to a hypertonic solution. In this case, the 30% sugar solution reduced the water potential outside the cells, causing water to move out through osmosis. As a result, the protoplast shrank and separated from the cell wall. This experiment illustrates the impact of osmotic pressure on plant cells and serves as a clear example of how cells respond to changes in the solute concentration of their surroundings.